Hello students, today we are going to study the basic reaction of polymerase chain reaction, that is PCR. PCR was invented by Caramelus in 1983 and this is an in vitro technique for the amplification of a DNA sequence. This reaction actually helps us to make a number of copies of the specified DNA sequence. Previously, as we have studied that a DNA can be amplified through recombinant DNA technology also, but this is another technique of molecular biology that aims for the amplification of DNA sequence. Now the basic reaction is divided into three steps. First is the denaturation step, which is done approximately at a temperature of 94 degrees Celsius. The second step is primer annealing, that is joining of the primers to the DNA strands, and the third step is extension, that is synthesis of the new strands of DNA. This is the diagrammatic representation of PCR. The very first step is denaturation. The second figure shows how denaturation is done. This is a double-stranded DNA that is heated to around 95 degrees Celsius and gives us two templates for the amplification reaction. Now we come to the second step, that is annealing step, in which the two primers, the forward primer and the reverse primer, attaches to the two template strands, and this is done at a relatively lower temperature. This is around 50 degrees Celsius, at which the primers bind to the template strand. The third step is the polymerization reaction step. In this figure, as you can see, how DNA polymerase is binding, and this shows the primer and the incoming DNTPs, and there is a synthesis of the new fragment. So this is the final figure which shows that this is template strand 1, this is template strand 2, this is the forward primer, this is the reverse primer. These are the two polymerase enzymes that are being shown, and this is how the DNA strands are synthesized. Now, how to calculate the number of molecules that we yield after this chain reaction? This is given by 2 raised to the power n, where n is the number of amplification cycle. So if we have n template copies at the beginning of the reaction, this goes into 2 with raised to the power n, that is number of amplification cycles. So this is how we can calculate how the number of DNA molecules that we will get after the specified number of cycles after the PCR reaction. In our next video, we will be studying the basic components of the reaction mixture and their importance. Thank you.